The bees have decided the queen is ready. The swarm cells are sealed. Swarming is about to begin. Most beekeepers panic at that point. They destroy swarm cells. They open the hive again and again. They move frames around. They look for a quick solution. But the problem remains, because the colony still wants to swarm. Once the bees have truly decided to leave the hive, it is often already too late, however. There is a method that is more than a century old. Simple, logical, and based on bee biology. A method that does not try to defeat the swarming instinct. Instead, it uses it. This is the Pagden Method, one of the most well-known swarm control methods in modern beekeeping. Today, we will explain how it works, why it works when it is used, what its advantages are, and why many beekeepers misunderstand its true mechanism. Because the Pagden Method is not just about moving hives, it is about changing the organization of the entire bee colony. Let's begin. The Pagden Method is a swarm control method. Its goal is not to eliminate the swarming instinct. Its goal is to change the conditions that led to the development of that instinct. When a colony reaches a critical population level, changes begin. Swarm cells appear. The queen reduces her egg-laying activity. The number of young bees increases. The internal organization of the colony changes. The queen is still present. The brood is still present. But the system begins preparing for colony reproduction. The Pagden Method uses the natural logic of that process. In practical terms, the beekeeper separates the old queen from the swarm cells, redirects the forager bees to the queen, and simulates the departure of a natural swarm without actually losing bees. The method is applied when the swarming impulse is already present. It is not a preventive measure. It is not hive expansion. It is not simply adding supers. It is a response to a colony that is already preparing to swarm. The most common signs are the presence of swarm cells, a large amount of sealed brood, reduced egg-laying by the queen, a large bee population, a congested brood nest, bees hanging outside the entrance. If swarm cells are simply destroyed at this stage, the problem often remains. The colony will usually build new ones because the cause has not been removed. To understand the Pagden Method, we must understand forager bees. Foragers possess an exceptionally precise orientation system. Throughout their lives, they memorize the location of the hive. They memorize the approach. They memorize the position of the entrance. They memorize landmarks. They do not search for the queen. They do not search for brood. They do not search for swarm cells. They return to the location they have memorized. In other words, they follow a memorized flight vector. That is the key to the entire method. If the beekeeper moves the original hive from its location, the foragers will return to the old position, regardless of where their previous hive is now located. The Pagden Method uses this biological characteristic, and that is exactly why it works. What happens before swarming? This is where we come to a point that many beekeepers overlook. Swarming is not a random event. Swarming is the result of changes in the colony's population structure. The number of young bees increases. The amount of open brood gradually declines. The need for larval care decreases. A surplus of young worker bees appears. The balance between brood and adult bees changes. The distribution of tasks changes. The behavior of the colony changes. In other words, the population balance becomes disrupted. This is one of the key factors driving the swarming impulse. And this is exactly where the Pagden Method intervenes. How is the Pagden Method performed? The first step is finding the queen. Without locating the queen, the method cannot be performed correctly. Once the queen is found, a second hive is prepared. The original hive is then moved several meters to the side. A new hive is placed in its original position. The queen is transferred into the new hive, along with several brood frames and a sufficient number of bees. Now comes the critical moment. The foragers return from the field. They come back to the location they remember. But the old hive is no longer there. Instead, the new hive containing the old queen occupies that position. Over the following hours and days, a large number of foragers enter that hive. As a result, the queen receives a substantial reinforcement of bees. In practical terms, she gains a colony structure very similar to a natural swarm. What happens in the new hive? This is where the most important biological effect occurs. It is not only the location that changes. The organization of the colony changes. The distribution of the workforce changes. The age structure of the population changes. The balance between nurse bees and foragers changes. A rapid redistribution of the population occurs. A large proportion of field bees now follows the queen. Nectar collection continues. Pollen collection continues. The colony continues to develop, but it no longer experiences the same conditions that were promoting swarming. That is why the swarming impulse begins to weaken. What happens in the old hive? The old hive remains with the swarm cells, but without a large portion of its foragers. That is a major change. The load on the system is reduced. Congestion is reduced. Task allocation changes. The internal dynamics of the colony change. A new queen develops from the remaining swarm cells. The colony continues its development through the process of generational replacement. The most common mistakes. The first mistake: the queen is not found. If she remains in the wrong hive, the method loses its purpose. The second mistake: leaving too many swarm cells. This can increase the likelihood of secondary swarms. The third mistake: reacting too late. If the swarm has already departed, the Pagden method no longer solves the primary problem. The fourth mistake: insufficient space for colony development. The fifth mistake: lack of follow-up inspection after performing the method. The Pagden method is not the end of the process. It is the beginning of a new colony state. If you want to understand bee biology, rather than just individual techniques, watch the playlist about honeybee colony biology. Watch the swarm management series. Watch the lessons on colony organization. Watch the content about communication and decision-making within the colony. Subscribe to the channel, because every lesson builds upon the previous one. It reduces the risk of losing a swarm. It uses the natural behavior of bees. It does not require complex equipment. It allows controlled colony management. It helps preserve the colony's production potential. It is based on colony biology. That is why it has survived for more than a century. It is important to understand that the Pagden method is not a magic solution. It does not eliminate the cause of swarming. Instead, it changes the conditions in which the colony exists. It does not eliminate biology. It uses biology. That is why it requires an understanding of the processes taking place inside the colony. Many beekeepers think the Pagden method is simply about moving hives, but that is not the essence of the method. The essence is changing the colony's population structure, changing the distribution of labor, changing the relationship between brood and adult bees, changing the flow of pheromonal signals throughout the colony. In other words, the organization of the system changes. And when the organization of a system changes, its behavior changes as well. That is why the Pagden method works. not because it tricks the bees, but because it uses the natural rules by which the colony functions. Whoever understands population dynamics also understands why the method produces results. And that is exactly where the true understanding of honeybee colony biology begins. If you found this lesson useful, subscribe to the channel and watch the complete swarm management series. In the playlists, you will find related lessons that together build a complete understanding of honeybee colony biology. In the next episode, we move on to one of the most fascinating methods in the history of beekeeping, the Taranov method, a method that uses a special board to allow bees to form an artificial swarm on their own. How does it work? Why does it work? And what is actually happening inside the mind of the bee colony during that process? That is what we will discuss in the next lesson. See you in the next episode.